Hello, welcome to a CNA's Opus. My name is Rich. I'm a certified nursing assistant. I apologize, I don't have much time to make these videos. But before we get started, first thing I need you to do is I need you to do this right here. I need you to do that right there. Because if you don't do that, then the rest of this is just nonsense or psychobabble. And I promise you that it isn't. I promise you it isn't. So moving on from there, this is a G protein coupled receptor, and that's where the chain of events is going to start. So G protein coupled receptors, again, are 7 membrane spanning alpha helices, and the amino terminus is on the extracellularly, and the C terminus is intercellularly. The G protein coupled receptor that has a high affinity to the S-alpha subunit of the heterotrimetric G proteins is B2 adrenoreceptor. So the ligand for this to start the chain of events for adenylyl cyclase is going to be adrenaline or noradrenaline. So epinephrine or norepinephrine. Those are going to be the two ligands that are going to come in here, make a conformational change intracellularly here to increase the affinity of the S-alpha family of heterotrimetric G proteins. See, we have the alpha, the beta, and the gamma, and it's always bound via lipid moiety to the gamma to the gamma subunit over here, and this is in its GDP state, so it's in its off state. So when adrenaline or norepinephrine comes in here and there's a conformational change, it'll bring this in and it'll turn this into GTP. Now remember, this beta-2 adrenoreceptor only kind of responds to this S-alpha family, which is either S-alpha or S-ulf. And it's only called ULF because it was found within the epithelial cells of the nose. But it's so similar to the S-alpha that they're really basically the same thing. And they're going to do the same thing. So this is S-alpha. And it's put into, once it binds to the G protein coupled receptor, it gets put into GTP, which is its on state. Okay? Once it's in its on state, it's going to go hit its target. Now, if you remember from heterotrimetric G proteins from the last video I made about phospholipase, yeah, it was phospholipase C. That was the target. The target in this one is Adenylyl Cyclase, which is an enzyme. It's going to break down adenosine triphosphate into cyclic adenosine monophosphate and pyrophosphate. So those are the two things it's going to break it down to. But it has specific, you notice it's going to hop from here, this G protein coupled receptor. This is going to, the ligand's going to bind. This is going to come over. It's going to get turned on, right? It's going to go to a GTP state, and then it's going to go look for its target. It's going to hit its target, and its target is adenylyl cyclase. Adenylyl cyclase looks a lot like the G protein coupled receptors, but it's different. That obviously it has this little loop here and all that kind of stuff. Now this is a six membrane spanning alpha helix, and that's one domain of adenylyl cyclase. Transmembrane domain 1 is this, and you'll notice too the amino terminus starts intercellularly. Then there's this loop right here in between that and this over here is another cluster of 6 membrane spanning alpha helix, and that's transmembrane domain 2 of adenylyl cyclase. So we have TM1 and TM2 over here. Then in the center here, we have this loop here is another domain called C1. And these are important. These are important. This tail here before the C terminus down here that is C2. So you have C1 and C2, but they're split up into subunits. What happens when this here, this S-alpha GTP comes over here and it gets kind of wedged in between here, it's going to loop this around, this linear portion around, and C2A is going to bind with C1A. So there's a dimerization. It's almost like a clamp, like a jaw shutting, almost like a jaw shutting, right? Boom. Almost like it could break things apart, like here. That is adenosine triphosphate. So adenosine triphosphate, you have the oxygen on the top right here. That's adenine. Now, adenosine just means the organic base adenine bound to the ribose sugar. And the ribose sugar has the two alcohol groups coming off of it, off the second prime and the third prime, which I don't show. So when it gets broken down into cyclic adenosine monophosphate, the fifth carbon and the third prime, via the alcohol group that's there on the ribose, those will come together. Oh, I didn't draw the adenine right here. Sorry about that, but there's still the adenine there. But remember, adenine is just adenine, but when you add ribose to it, it's adenosine, and then obviously monophosphate here and triphosphate up here for the three that's coming off. Then this, what's left over, what was kind of clipped off right here, is pyrophosphate. So those are the two things that you're left with, is cyclic adenosine monophosphate and pyrophosphate after like the jaw shots here, and there's a dimerization of the C2A and C1A, and that's after, you know, C alpha in its on state comes over and creates that reaction. It's a little bit more complicated than that, but we're gonna keep it cool. We're just gonna keep it cool right now. So anyway, so here's your S alpha, and I think I went over this before, but you could look at this as just the G monomeric protein by itself, like RAS. So here's there's a cysteine residue right here coming off, like coming out of it. There's a little cysteine that could be attached to, and the cysteine attaches to a fatty acid right here called palmitic acid or hexadecanoic acid. So we know there's going to be 16 carbons, hexadecanoic, and then the acid here. So there's 14 carbons. It's fully saturated. So it has there's these 14 methylene groups right here going down. And then there's a methyl group on the end here, because that has the third, just to show that it has a third hydrogen, and the carbon has all four bonds. But deca is that's 10, and hexa, that's 6. So we have 16 noic acid, carboxylic acid, fully saturated. And then the cysteine down here, that I wrote as a residue, meaning it's connected to another amino acid. So the nitrogen only has the one hydrogen coming off of it, and that's connected to the carbonyl group of the next amino acid via like an amide link, which you'll see in a second. That's the carbonyl group right there that it would attach to the next one up here. So this right here, the next amino acid up there will be right here, and that would be an amide link right there. But right over here, this has the cysteine has a thiol group off its R group right here. It's a methylene group, and then the thiol group right here. And the thiol group is very similar to an alcohol group, but instead of an oxygen, it's a sulfur. So what happens is, how they react is the alcohol group right here comes together with the hydrogen that's next to the sulfur, and it's a condensation reaction. So water is released, and the sulfur bonds with the carbon. So the carbon has its 4 links, 1, 2, 3, 4. So that's four. This is 4 links, and then this up here is a fatty acid, so it's extremely hydrophobic because the electronegativity of the carbon between the carbon and the hydrogen is relatively the same, and the electronegativity between the carbon and the carbon is the same, meaning there's no pull. The protons that are in here aren't swaying the electrons from the hydrogen over to this direction, making it more negative and this more positive. It's neutral. The water molecules, which I should have shown, are acting like crazy. They're making intercellular links to kind of stabilize themselves. And hydrogen bonds between the oxygens, and some of them get partially negative and some of them get partially positive. And oxygen has 2 lone pair electrons, so it's already in a negative state as is. But it has a very, very difficult time interacting with something that's neutral. It wants to react with something it can latch onto and stabilize itself onto. And this is already stable. It's already holding itself together. So the oxygen will just boom, boom, away from it. And fatty acids will aggregate together. And the one place it's going to aggregate together, obviously, is in the phospholipid bilayer, because that's where all the fat is. It's right there. So it'll latch on there via the cysteine, and then it's not that the fat's reacting with one another, it's that the water is reacting with one another, and it's just driving that up to where there's neutrality, which is in the cell membrane. So I hope that gives you a little bit of intuition on what I'm saying, and a little bit of an idea. I should have wrote it out. Maybe I will in another video. But moving on from there, there's a special one in these in that heterotrimetric G protein coupled receptors, and that's the G alpha I NOR family. And that one has the cysteine residue, so that attached via palmitic acid. Actually, it doesn't bind via the acid. What's left there bound to it is the acyl group, because the alcohol is gone now. So this is just the acyl group that it attached to, and it has a specific name. I just forget what it is. So this is myristic acid over here, or to make it easier, it's tetradecanoic acid. So that's 14 carbons. So we have 12 methylene groups, then the 1 methyl group right here, and then the carboxylic acid right there. And again, it's going to be the same reaction, but this only happens. This link with this family of the G monomeric proteins in the heterotrimetric G proteins. I know that's a mouthful, but this one right here is going to have a very, very strong link to the phospholipid bilayer. It's going to be extra strong. It's going to be extra stable there, because it's going to create another link that the other ones aren't. And the reason that is the start codon that comes out, the first amino acid that comes out in proteins is methionine. Is methionine. That's the start codon, is methionine. But a lot of times that gets clipped off. And if the first amino acid in it after it's clipped off is glycine, which happens in this family here, there'll be a reaction here. So this is the glycine is now the amino terminus. See, it has the nitrogen with the 2 hydrogens instead of just 1 hydrogen. It has 2. This is the amino terminus. There's no other amino acid next to it right now. Pretend there's no amino. Just block this out. Just block that out. There's no amino acid next to it. Okay? So it has its 2 hydrogens. Now, we're going to link something onto it. So one of the hydrogens is going to disappear, just like in the same other reactions. This only happens when glycine is the first amino acid in the link. And I have an intuition as to why that is, but I don't want to kick it out there. I think it has but I'll kick it out there anyway. I think it has a lot to do with in the R group of glycine. The only thing in the R group of the glycine, or the functional group, is a hydrogen. Okay, so off the alpha carbon right here, the only thing coming off is a hydrogen, nothing else. So that makes it a little bit it gives it a variation different than other amino acids. It's very simple. So it forms this. This is an amide link, just like a link that would be made between two amino acids in a polypeptide or in a protein. So this alcohol group off of the carboxylic acid will make a link with the hydrogen right there, and that'll form water. So that'll be a condensation reaction. So then you'll have the carbonyl right there, and then the nitrogen and the hydrogen right there, and that's an amide link. That's a link that's made between amino acids within proteins, but this is it can make it to the fatty acids too. And that's what this is. And again, it makes a link to the fatty acids. So the oxygens are going to go their separate route away from it. And because it's hydrophobic, and I hope I give you a good intuition as to why it's hydrophobic. It's neutral here. So now you have two links in this family. You have one from the glycine, because the glycine is the amino terminus, and then you have one like the rest of them have at the cysteine, and then because it's a heterotrimetric G protein, you have the beta and the gamma subunit, and it's always bound via lipid moiety over here on the gamma subunit, and not on the beta. And I know how this reaction happens too, but it gets crazy and hairy, and I'm not gonna do it. I mean, I can do it, but this video will go on for another 20 minutes. And that right there is going to be it for right now. I just want to show those reactions right there because I think they're pretty cool. But I'm going to make another video on what happens as the saga continues with adenylyl cyclase and protein kinase A. Protein kinase A. Because last video I did protein kinase C. So this will be protein kinase A. And cha-ding, that dimerization, and that turned the adenosine triphosphate into cyclic adenosine monophosphate and pyrophosphate. You're gonna see that there's a binding site, cyclic AMP, which this is going to go up in the cell membrane near the phospholipid bilayer, and there's a link. There's binding sites for it on protein kinase A, and then we're going to go from there. All right. October is Breast Cancer Awareness Month, so I will be doing a full video, just in one video, on breast cancer. And that is it. Rich, CNA. See you later.